So while Rich was preparing much of today's and tomorrow's content, I was actually on a 3-week vacation. In the Sierras in California. So I'm spending a bit too much time in California right now. But truly a massive kudos to Rich in his presentation. Okay, so I work on the development of some of the algorithms used in CryoSPARC for 3D reconstruction. I originally joined the team back in 2020 as an undergrad intern. And back then I worked on some of these tweaks to the standard workflow that Rich presented. Tweaks to help CryoSPARC work a bit better for proteins with helical symmetry, actually. And since that project, I've worked on various other topics in 3D reconstruction, focusing a bit on geometry and symmetry. And so our case studies today won't actually cover helical processing, but feel free to approach me later today or tomorrow if you have any questions about helical processing in CryoSPARC. Okay, so if you couldn't tell, the theme for today is definitely one of that of symmetry. And tomorrow we'll really kind of dive into heterogeneity, and Rich will take over again. The case study that I'll cover today is going to focus on the structure of encapsulated ferritin. And this data set presents a significant challenge, mainly due to its unique geometry and symmetry. And it requires some advanced tools in CryoSPARC to obtain a high resolution structure. And so while we're processing this data set, we'll be taking on a journey involving some jobs like local refinement, like Rich demonstrated earlier today. And we'll also talk about particle subtraction, which is a kind of auxiliary tool to local refinement that you can use. And we'll as well use Align3D, Regroup 3D, volume alignment tools, a lot of these jobs that Rich was using this morning. Yeah. We will also explore the concepts of local symmetry and non-point group symmetry. And we'll talk about how a practitioner might go about handling these in CryoSPARC. And keeping with the theme, we'll explore some processing steps that seem intuitive and that I certainly thought would be worth trying, but they actually didn't work very well. And we'll try to build some intuition as to why they didn't work very well. And this is definitely a case study where multiple processing strategies would have been viable for sure, as pretty much every case study is. Yeah, so why don't we get started? And before I kind of launch into things, I'm just going to cover some very basic terms concerning symmetry in CryoEM, just to help contextualize the talk. And some of these will be reviewed from earlier today for sure. But I use a few of these terms throughout the case study, so I just want to touch on them right now. Okay, so in the context of CryoEM, typically we call the asymmetric unit the smallest region of the molecule, which can be used to generate the complete structure together with the symmetry group. And of course, this only applies to symmetric molecules. I'm sure many of us who have gone through CryoSPARC introductory tutorials are familiar with the T20S proteasome dataset. And this dataset has an asymmetric unit comprising of both the alpha and beta subunits. Those are shown in purple and gray. And these comprise 1/14 of the full proteasome structure. Also point group symmetry, very basic, but we use that to refer to the set of rotations that when applied to an asymmetric unit, a symmetric structure, leave it unchanged. And so they, by definition, keep the origin fixed in space, hence the name point group or fixed point. And so in CryoEM, the common point group symmetries that are implemented in most softwares, such as CryoSPARC, are cyclic groups, like the trip channel, also dihedral symmetry, and then higher order symmetries like tetrahedral, octahedral, and icosahedral. Okay, great. So for the purposes of today, I'll use the term non-point group symmetry to refer to any form of self-similarity within a molecule that doesn't fit into the concept of a point group symmetry. And so one example of this in CryoSPARC is, of course, helical symmetry. The reason why it's not a point group is because it combines a rotation around the helical axis with a translation along that helical axis. So it doesn't leave any point in space fixed. And another example is local symmetry. And so this is where the symmetry is restricted to only a small, spatially compact region of the molecule. And encapsulated ferritin will be one example of this local symmetry. And then pseudosymmetry, which Rich talked about a bit earlier today, generally refers to when only approximate symmetry exists in the structure. And so, for example, the symmetry might only apply to some, but not all, of the asymmetric units. And this is actually what is happening with the trip channel and calmodulin. So calmodulin is only binding to one of the binding sites, and it makes that asymmetric unit different from the other three, which are identical. And another example is when the symmetry is disrupted by flexibility. Okay, so a little bit of review as well from earlier today, but I do want to touch on why it's important that we even think about symmetry in cryo-EM in the first place. As Rich pointed out, symmetry gives the opportunity for reconstruction algorithms to have much better performance. And it does this primarily by boosting the number of observations in the data set. And so, for example, if I had a data set of 1,000 particles of the T20S proteasome, that's just 1,000 projections of the structure. However, if we know that the molecule is symmetric, for example, in this case, we know that it has D7 symmetry, though it still contains 1,000 projections of the full molecule, it contains 14,000 projections of the asymmetric unit. And so that's a 14-fold increase in the number of data and the number of projections that we have to work with. Okay, so in figure A here, the full T20S particles are shown in the micrograph, and they're circled with green circles here. And then on figure B on the right, there's a green dot over each of those 14 asymmetric units, one for each particle picked in figure A. So 14 times the number of green circles in figure B than in figure A. And of course, taking advantage of these extra projections can give CryoSPARC more data to work with. So it's definitely going to result in an increase in your resolutions. Now, symmetry also provides a strong source of prior information about the relative poses within a structure. And specifically, knowledge of symmetry tells us the relative position and orientation of the asymmetric unit. Now, something important to note on, and that we'll encounter today, is that symmetry doesn't always make things easier. In fact, it usually makes things more tricky. And one way that it does this is it creates difficulties for reconstruction algorithms, or it can create difficulty with reconstruction algorithms when it's not accounted for. And so this is an example of running ab initio reconstruction on an apoferritin data set without enforcing symmetry. So here, imposing symmetry in figure B on the right is actually necessary to obtain a reasonable low resolution result. And apoferritin obviously has octahedral symmetry, which is a 24-order symmetry group. And this is just one particular pitfall of ab initio that you may encounter if you're working with high symmetry data, particularly viruses are particularly susceptible to this, as well as helical proteins. Okay, so it's still the case that we can't just ignore the symmetry in a structure. And knowing it can both boost the number of observations, the quality of our reconstructions, can increase the quality, and also help us avoid this kind of pitfall that we encounter with ab initio reconstruction in particular. Okay, so now on to today's data set. The data set that I'll be focusing on is an Encapsulin nanocompartment. And this data set was originally collected by Jennifer Ross, as well as her colleagues in 2022. And this contains 4 encapsulated ferritin decamers within. So I'm not a life scientist, but I do highly recommend reading this paper published by Ross et al. For more biological information about the sample. It's a super interesting data set, as we'll see. And in this paper, they present a 2.2 angstrom resolution structure of encapsulin and a lower resolution structure of the encapsulated ferritin that's inside. And so a screen cap of the density is shown here on the right side. The density here is colored radially. So encapsulin is shown in magenta, and it's located along the outside. And the 4 copies of encapsulated ferritin are teal, and they're located inside the structure. And note that this is not their final or highest resolution structure. We'll show that a bit later. Today, we'll show how a practitioner might go about processing this data set to reconstruct both the encapsulin and the encapsulated ferritin. And doing so, we'll actually be able to get a high resolution structure of the encapsulated ferritin, higher than what we've seen before. Okay, so just a little bit of background on the biology, or the sample that we're working with, not really the biology. So encapsulins are a type of protein that contain internal cargo. This encapsulated encapsulin is an icosahedrally symmetric molecule. It's from the species of bacteria, which I won't attempt to pronounce. And encapsulin nanocompartments are hollow, and they exist in prokaryotes. And they range in size from about 20 to 40 nanometers. And we're calling the contents inside their cargo. And typically they contain enzymes. So in this study, we're studying an encapsulin that contains 4 copies of a ferritin-like protein, so hence the name encapsulated ferritin. Okay, so the geometry of the nano compartment is best illustrated by this figure here. Okay, so starting from the left side, the outer shell is displayed on the left in blue, and that's encapsulin. It has global icosahedral symmetry, so fairly straightforward. And inside are 4 copies of encapsulated ferritin. The abbreviation is EncFTN, but I'll just say encapsulated ferritin. And these 4 are arranged in an approximate tetrahedral configuration. Each of these individual encapsulin ferritin molecules are themselves composed of a pentamer of dimers, is what we'll call it. And what that really means is it has D5 symmetry. So D5 is a 10-fold symmetry group. So there's actually 10 copies of each asymmetric unit inside each of these encapsulated ferritin molecules. So since there are 4 copies of encapsulated ferritin inside the complex, each particle we're working with actually has 40 copies of the asymmetric unit for encapsulated ferritin. And it also has 60 copies of the asymmetric unit for the icosahedral shell, the encapsulin. Okay, so our goal with this data set will be to reconstruct both encapsulin and encapsulated ferritin. And as we will see, encapsulin is fairly straightforward to reconstruct, but the insides require some special tricks to process. And specifically, we'll need some custom steps to take care of the geometry, the custom geometry of the sample. So I'll walk through the steps we took to handle this geometry in CryoSPARC and how we got a final structure with this data set. Okay. Okay, so Rich introduced the standard workflow, or Rich's standard workflow from this morning, but I think it's fairly standard, so we'll call it that. The standard workflow that he presented works quite well for encapsulin. In this case, we completed pre-processing, so motion correction and CTF estimation for this data set. And then we did blob picking. Here, the only thing that I did that was maybe of note is in this case, we're working with a virus, and the data set looks quite a lot like the particles in the micrograph, rather, at very low resolution, look kind of like rings or circles. So in this case, it's like the kind of perfect example for when you might want to use the ring-shaped blob in the blob picker. And that's what I did, and it worked pretty well for particle picking here. After that, I did some coarse particle curation, and this is via the inspect particle picks job, mostly just removing very either noisy picks that were just pure noise and like false positives, as well as removing some ice picks. After that, I did template picking, so I took the good particles from that, ran through the classification and got some templates, and then I went back, did template picking again, and then had a cleaner set of particles to work with. And then I did thorough particle curation, as Rich described, which was just one round of select 2D classification and select 2D classes. In this case, because the data was quite good, and though there's relatively low, minimal conformational heterogeneity here, I was kind of satisfied with just using curation in 2D at the start. And we will see later on that this wasn't sufficient for 3D, at least for the 3D of encapsulated ferritin. And these steps work mostly out of the box, so I won't go into these in too much detail. Okay, the next step was, how do we get an initial model? And this is where the first kind of gotcha came up. Okay, so now that we have our set of curated particles, we want to move on to initial model generation. And as I kind of alluded to a bit before, those of you with experience working with virus or helical data sets, anything with high symmetry groups, a common kind of pitfall of ab initio reconstruction, but refinement algorithms in general, is where they will separate more on orientation and less on heterogeneity. And even in the case of a single class, they can often put all particles in a very similar orientation. And this failure mode is somewhat particular to ab initio reconstruction. Intuitively, we think that this arises because particle images of high symmetry data sets look very similar to each other when they're low pass filtered to a low resolution. And so since ab initio reconstruction by default only works with low resolution data, the algorithm finds it easier to place all particles with the same viewing direction. And so you can see this via the relative signal amount plot over orientations. It's putting most particles along an azimuth and elevation of both 0 and -pi. And the elevation, there's like no particles with top-down views. It's all horizontal. And you can see in the volume, of course, it means along one view, it looks a bit like a 2D projection. Along the other view, it does not look like anything that we expect. And this pathology was actually initially reported on our discussion forum by users like you. So another plug for, we love, every time we see things on the discussion forum, we promise we take note of it and we triage it internally. So yeah, great place for sharing results and sharing discussion about any sort of weird things that come up. Okay, so there's two options that we can kind of employ to work around this issue, or at least two options that I'm kind of aware of. The first is just to enforce symmetry during ab initio reconstruction. This will, of course, guarantee that a symmetric structure is found, as we expect. The other option that we kind of discovered recently that seems to work well for this case is turning off non-negativity. And so we don't really know why this works. This is one of those black box moments where even us are a little bit fooled. But this parameter has been found to discourage ab initio from producing flattened models. And this was discovered by one of our researchers, Valentin. Okay, I'm going to do a think pair as well now here. I want you guys to think, what are the disadvantages of enforcing symmetry during ab initio reconstruction? And if we later want to resolve an asymmetric feature in a data set, is it in our benefit to enforce symmetry from the start? And this is quite a bit of review from this morning, so I'll probably kind of not do the full 4 minutes of discussion. And it will be a bit more low tech than Rich's. I'll simply call out when it's time to share with your neighbor. But think about it for about a minute, and then I'll call out. All right, so for the next 3 minutes, discuss this with your neighbor. All right, so let's bring it back in. We're really getting you guys to think a lot about symmetry today. Um, Still about an hour and a half more of that, unfortunately. No, I'm kidding. Okay, so so enforcing symmetry is undesirable, as we kind of talked about this morning, because in this case, we would actually like to preserve the internal asymmetric structure within the encapsulin as best as possible. Specifically, this internal, I'll just go back a little bit, this internal arrangement of the encapsulated ferritin definitely does not follow icosahedral symmetry. So if we were to enforce icosahedral symmetry, it would disappear. One thing to note is that it's actually not always the case that enforcing symmetry during ab initio reconstruction is undesirable. And some of the time it might actually be necessary. For example, we might not initially expect there to be any symmetry breaking features in a data set that we're working with. And we might want to process the entire data set with symmetry enforced all the way until refinement. Then once we have a high resolution consensus structure, we could use tools like 3D classification or 3D variability to reveal some of these symmetry breaking features. And this approach is sensible to us when symmetry breaking features are not large scale. Typically when you're looking at medium or high resolution changes or very small changes to a very small region within your structure. So in our case, we fall more into the former case. The symmetry break is large scale. The entire internal contents of encapsulin are symmetry breaking. And so we have most success just breaking the symmetry right from the start. Yeah. Okay. So for this data set, we went with not enforcing symmetry, and we used the following parameters for ab initio reconstruction. So enforced non-negativity was off. And we also turned maximum resolution and initial resolution. We both bumped these up, or the numbers down, to 8 angstroms and 25 angstroms, respectively. And I increased both of these because I wanted ab initio reconstruction specifically to focus on medium and higher resolutions right from the beginning. And this is because at low resolution, all these particles look basically like circles or like rings as we used in blob picking. So nudging these values up a bit just helps give ab initio a bit more information to work with right from the start. Okay, using these parameters, we actually nicely recover both the encapsulin as well as the internal tetrahedral configuration of the encapsulated ferritin, shown here in the bottom left. And after this, I ran 2 refinements because I wanted to see what would happen. First, I ran an asymmetric refinement with just C1 symmetry, which kind of just looks like a higher resolution structure of this now. But the nice thing is that we didn't lose the symmetry break. And then I also ran a symmetry enforced refinement. So this is an icosahedral symmetry refinement. As you can see, the C1 refinement preserves the internal cargo, which is present, but still at low resolution, still very blurry and noisy. But the icosahedral enforced refinement, it destroys the signal of the cargo entirely. It's basically absent in this volume, which I've thresholded out. But the structure of the encapsulin is notably higher resolution. In this case, it's actually using about 60 times, exactly 60 times the number of particles as the top one. And in both refinements, I also disabled dynamic masking. And this is something that we have started to experiment with a bit. Dynamic masking is by default kind of employed in the refinement jobs to help remove signal outside of what we think the structure is. The problem is when you're looking for symmetry breaks, you are probably interested in areas of your map that are initially low density or have lower density values. And so if you want to be as careful as possible, what we often suggest is disable dynamic masking and use a static mask or even no mask entirely, just at the start to see first if you can keep the refinement as minimally biased as possible. Okay. So great, we now have a structure of encapsulin, and it wasn't that painful, resolved to fairly high resolution. And we have both C1 and icosahedral structures of it. Awesome. The next step that I wanted to do was to reconstruct the interior, the encapsulated ferritin. So somehow we have to tell CryoSPARC to focus on the interior. And typically, this is done with a local refinement job with a mask covering the portion of interest. Before we attempt local refinement, though, what I wanted to do first was to remove the encapsulin signal from the images. So how do we do this? This is done with the particle subtraction job. Okay, Particle subtraction takes as input the particle images, obviously, and these must have been aligned previously to this structure. It also takes the associated volume that they were aligned to, and it takes a mask covering the region to subtract. Now, how can we make this mask? Well, we'd like to subtract Encapsulin, so we want the mask to cover only this region and to exclude the interior of the nanocompartment. So typically, we'd have to use some tools in UCSF Chimera X, like the volume eraser, or perhaps the map segmentation feature to generate such a mask. However, in this case, since Encapsulin is the dominant signal in the volume, we can actually just increase the threshold to remove the interior of the nanocompartment. And this works pretty well. Then we can generate a mask of just the Encapsulin using the volume tools job in the mask output mode. So this was convenient, but it doesn't always work out this way for sure, especially if you're erasing different regions of your map or the area you're interested in is not just low density or just high density. Okay, so after giving the particles, volume, and the mask to the particle subtraction job, the output is images with Encapsulin subtracted. And so this is just a GIF flashing between particles before and after subtraction. And you can see that in most of these images, there is something in the center. I'll just replay that a bit. There is something in the center that actually is the internal contents that we're interested in. And it's not super small either, which is a good sign. One thing to note about particle subtraction is it's typically considered an optional step before local refinement. Sometimes it helps. In this case, it did help, but other times it's neutral or it can even make results worse. And this is, again, data set dependent, unfortunately. The reason is that there's many variables at play. It's not just the size of the area that you're subtracting away that is going to matter. It's also how accurately have you aligned your particles before then. So if you've gotten a lower-ish resolution structure and you'd like to use particle subtraction, that can impact the quality of the subtracted images because the alignments weren't that good, the volume was a bit lower resolution. So it's something to think about for sure. This is why it's kind of another case where it's worth it to try both with subtraction and without if you are experiencing difficulties with either method. Okay, great. So now we have our subtracted particles. The rest of this case study will focus on taking these particles and reconstructing encapsulated ferritin. And so we'll go through a few processing strategies and explore some steps that didn't work. And we'll also see some extra steps that I needed to take in order to avoid getting overfit maps or avoid getting stuck at lower resolution and end up with some high resolution structures, or just one structure really. Now I want to talk about two jobs that I first tried that would probably be the obvious approach to do at the beginning and why they didn't work. The first thing that I tried after I got these subtracted particles was just a straightforward homogeneous refinement of all four decamers through the homogeneous refinement job. In this case, I carried out the refinement without a mask. Since the Encapsulin was already subtracted away, I kind of didn't really need a mask in this case. And I also wanted to limit the mask bias as much as possible. So unfortunately, this refinement didn't reach a super high resolution. It got stuck at about 10 angstroms. And you can see on the bottom right, the image in salmon, that was kind of where we were at this point. And I thought, okay, what might this reveal about the sample, or what might this reveal about the data set at this point? Like, why weren't we getting to decent resolution? Okay, so each encapsulated ferritin unit is about 60 kilodaltons, something like that. So for these together, about 240 kilodaltons. This is quite a bit above the lower limit of molecular mass that we'd expect to be able to refine for a well-behaved sample, whether local refinement or homogeneous refinement. At a mass of 240 kilodaltons, refinement should be able to get a decent resolution, assuming that the particle images actually obey the forward model that Rich introduced. That is, assuming that the particle images are actually projections of a static structure. And so to me, this implies that the issue is not one of alignability, but more likely one of heterogeneity. So in this case, there's kind of two ways that could manifest. One way is the tetrahedral arrangement of the subunits not being the same in each image. So in this case, maybe the tetrahedron is like not exact. Maybe it's like wiggling around a bit in each image. And in this, I kind of will call this, I guess, inter-subunit heterogeneity. The other way it could manifest is conformational heterogeneity within each subunit itself. And I'll call that like intra-subunit heterogeneity. At this point, I wasn't really sure what it was. Okay, the second approach that I tried was a local refinement of just one encapsulated ferritin subunit. So I arbitrarily picked one of these four, and I put a mask around it. And then I used a local refinement job. So here, now we're using a mask, now we're using local refinement, and we're also using a much smaller region of our protein than before. So this kind of opens up the landscape, or opened up the possibility of overfitting when things don't work out well. So this is kind of the problem that we ran into here. So instead of being stuck below 10 angstroms, we got a claim resolution closer to about 7 angstroms. But the map shows quite a bit of overfitting kind of artifacts. And these are shown in the figures in the bottom right. The map at a high threshold shows that the strongest density regions are mostly just streaks kind of going out radially. And Rich kind of said in both 2D classification and in 3D, you'll see oftentimes these radial streaks. They're very telltale sign of overfitting. And if you see them in your map, it's very likely artifactual and not real. These artifacts are consistent with our experience when there's too many junk particles in the data set. It's also consistent of our experience when there's still residual heterogeneity. For example, in this case, intra-subunit heterogeneity within a singular encapsulated ferritin that's unresolved. And thus local refinement takes these junk particles and can use them now as an extra kind of knob to an extra variable to push around to, in this case, overfit. This can also happen though when the initial alignments are too far off from their optimal values. So that's where the search extents and local refinements are kind of important to think about. Okay, so if local refinement fails and also if homogeneous refinement fails, what can we do instead? And based on kind of what we talked about this morning, there's probably a few ideas you have. Before we kind of answer that question, I thought we could take a brief break for questions, if anyone has any. Yes. If no one has any, we can also. Okay.
In the structure is
Yes.
are the fivefold axis of the ferritin aligned with the fivefold axis of the encapsulin?
Yes, so that's a great question. No, it is not. And in fact, that is what makes this kind of really tricky. So I don't know off the top of my head how many fivefold axes are in an icosahedron. Maybe it's
12.
Okay, there we go. In this case, I initially hoped that was the case when I was processing this, but no, they weren't. And so I had to use, in this case, well, we'll go into it. But yeah, it's a good question.
Are the fivefold axis of the ferritin aligned with the threefold axis of the encapsulin?
No. So they're actually, I'm pretty sure they're not aligned with any axis of the icosahedron. Yeah, I should. Yeah, so I couldn't go too in depth in this because icosahedron has 5, 3, and 2. Here, what I initially tried to do was tetrahedral processing of this, but we'll go into why that didn't work in a bit. But yeah, like based on what I tried, it doesn't align with any of the icosahedral symmetry axes. Okay. Oops, sorry. Here. Okay. I will head on. And before I kind of answer this question about why did local refinement struggle here and what can we do instead to get further, I just want to talk a little bit about 3 jobs that are used for volume alignment in CryoSPARC. And some of this we did discuss this morning, but it would be nice to review before we use them in kind of in gory detail. So the first job that I'm going to use today is Align 3D. And so as Rich showed, it's possible to end up with volumes that are out of register as outputs from ab initio or from heterogeneous refinement, or even from a 3D classification job in some situations, as we'll see later today. And what the Align 3D job will do is it will search rotation, it will search over 3D rotation and 3D shift, and also chirality. and it will align an arbitrary number of volumes to a reference map. So the output of this job is each volume now aligned to the reference map and written out as a separate output. So this is useful as a post-processing step for heterogeneous refinement, 3D classification, and it's also used for when you want to put your maps in register to perhaps your best refined structure currently from a homogeneous refinement, for example. Align3D will also accept the masks and the particles associated with each volume, and will put these into register based on the transformed volume to volume alignments. And this detail is very important because what it allows us to do is take particles as a group and realign them based on aligning their corresponding volumes. And as Rich said, particles, it's very, very difficult to work with them on an individual particle basis unless we have very, very well crafted algorithms. For aligning 3D maps, things can be quite a bit easier. And so if we don't need to kind of work on an per particle basis, it's nice to have these tools that we can just apply on the scale of a whole volume. And so here's an example of the outputs from a heterogeneous refinement. This is a respiratory complex 1 data set, EncFer entry 10927. And this is actually solved by one of our application scientists, Hannah. And in this case, ab initio was run, and it produced 3 coherent classes as well as 2 junk classes, which are not shown here. However, the 3 coherent classes ending up kind of resembling each other very strongly. They were basically just out of register. So compared to the first map, the second map needed a rotation and a shift to be aligned, and the third map needed all of those plus a hand flip or a chirality flip to be aligned. And so previously, we would have to correct this manually at the end of processing. But now Align3D can handle all of this. And in CryoSPARC, we updated Align3D in version 4.5. And it used a bit of a more robust alignment algorithm and also some memory improvements as well. OK, the next job that we are going to use a little bit today is Regroup3D. So Regroup3D is a new job as of CryoSPARC version 4.4. And what it does is it reclusters particles or volumes, really, from an upstream classification job into a smaller set of superclasses. And it does this using a fairly lightweight 3D voxel-based spectral clustering approach. And so what it does is it takes inputs from 3D classification, specifically the volume inputs, the volume series, and it takes the desired number of superclasses, which must be less than the number of classes that you ran 3D classification with. So in this example, this is the same respiratory complex 1 from before. We have 5 initial input classes, and here the first, here the 5 input classes, the first 3 are the similar ones that I showed from the previous slide, and the bottom 2 are quite different. And here I kind of looked by eye and saw, okay, these 3 classes look very, very similar. The other 2 don't really look like each other, nor do they look like the initial group. So what I wanted was 3 superclasses of reorganized volumes. So I told it, okay, let's give us 3 superclasses. And what the job did was then organize each of these volumes into a new set of output superclasses based on pairwise similarity. And so similarity here is measured by the Pearson correlation coefficient of these volumes. The job outputs 1 volume for each of these 3 superclasses, and that volume comprises of the average of the volumes that contributed to the superclass. So in the case of the first 3 classes that were all very similar, the average superclass is shown in the bottom in purple, and that's one of the outputs of this job. OK, we updated this job in CryoSPARC version 4.5 as well. So before version 4.5, Regroup3D would assume that all the volumes are in register already or in alignment already. But now in CryoSPARC version 4.5, Regroup3D will optionally compute the best alignments between all pairs of volumes, and it will use these alignments to estimate the correlation between each of these classes. So every pair of volume will be compared to find their optimal alignment, and then the Pearson correlation coefficient will be computed at that optimal alignment. Then it will output average structures between all volumes in each superclass. OK, great. The last job that I'll touch on, and pretty quickly, is just volume alignment tools. So it's a utility job that in CryoSPARC, and we saw it earlier today as well. And it comprises some low-level tools to adjust the position and orientation of volumes, masks, and particles in 3D. And we're going to use this a lot today. So the main 2 operations that this job can perform is aligning a volume to a symmetry axis. And this is kind of just a GIF of what an example looks like of an unaligned versus an aligned volume kind of doesn't really mean much because you can't see the symmetry axes here. And the other function that this job can do is it can recenter and rotate a volume arbitrarily. And we'll make use of this later today for helping to superimpose all of the 4 encapsulated ferritin molecules. Okay, now we'll head back to the case study. So before this interlude, we had faced a little bit of difficulty with using local refinement. It seemed to result in some overfitting, which we suspected could be due to alignments being too far off, or due to heterogeneity or junk. We weren't really sure. And we didn't know what we could do instead. There were also a few potential issues that we hypothesized were causing this. Junk particles being an obvious one. So instead of local refinement, what I turned to next was 3D classification. So 3D classification is nice because, unlike a refinement, is actually able to do something about these junk particles and the unresolved heterogeneity. Interestingly enough, though, one of the kind of findings of this case study was that 3D classification can also do something about poor alignments. Specifically, 3D classification revealed that within the initial particle alignments, there was actually a significant amount of diversity in the position of this tetrahedron arrangement relative to the encapsulin shell. And so here I'm showing the volume series of the classes that came out of 3D classification. Those are shown in gray on the right side. We'll just replay that as well. Looking at this volume series, we can see that the volumes are not in total alignment. Especially towards the end here, the tetrahedron is kind of jiggling around quite a bit. And using 3D classification, it spent quite a bit of its capacity in finding similar volumes oriented slightly differently relative to each other. Now, this isn't surprising because we haven't actually updated these alignments since that initial C1 refinement of the entire encapsulin and encapsulated ferritin complex. So kind of it's been a while, and we tried the local refinements, but they suffered from the overfitting issues. So we can actually, the next thing that we can do that was kind of unique about this case study is we can put the volumes back into register, as well as update the particle alignments using the Align 3D Maps job. Okay, I'll play this one more time. In yellow or in orange is the Align 3D classes after running Align 3D. So you can see that they're quite a bit more spatially coherent. And then also I'll show a static image here of all of them overlaid on each other. So this technique of using 3D classification to resolve first orientation diversity and then using Align 3D to put volumes back into register is what I like to call group realignment in this case study. So what are the advantages of doing this? Well, as we saw, 3D classification is, in this case, it's less prone to overfitting than local refinement, specifically because A, alignments are frozen, thus they're never estimated on a per particle basis, and B, it can create new classes, or rather it has more classes to work with. It doesn't have to force all particles to look like the same structure. Heterogeneous refinement and local refinement both attempt to estimate alignments on a per particle basis, and that can be problematic when there's still quite a bit of junk, and also when alignments are far from their true or optimal values. Now, the fact that these classes are out of alignment tells us something interesting about the sample, which is that the orientation of this tetrahedron relative to the encapsulin is not constant across particles. So that we suspected this earlier from the local refinements that we saw and the homogeneous refinement that didn't get to high resolution. But this is sort of more concrete, a little bit more concrete confirmation of it. And this also kind of ties into why the position of the tetrahedron relative to the icosahedral symmetry axes is so poorly defined. It doesn't seem to be constant at all based on the 3D classes we got in gray. There's just so much discrepancy in the orientation of these encapsulated ferritin. And keep in mind, these alignments are from the aligned encapsulin structure. So we would expect if there's any coherent detail aligned to that structure, it should show up here. Okay, great. So from this, we now have a single structure of the tetrahedron. After we've used Align 3D to put these volumes back into register, we did a reconstruction only. And that gave us this, this gray structure here. So we're still kind of at fairly low resolution, unfortunately. The next portion of this case study describes how CryoSPARC can be used to handle non-point group symmetry. So we have a few options here. We could, as we attempted before, just mask out one of these encapsulated ferritin subunits and attempt to refine it. That's option one. However, if we don't attempt to incorporate knowledge of the fact that the other three subunits are expected to be identical to that one, we'd effectively be throwing away about 75% of our data. Exactly 75. Well, some particles maybe don't have all 4 of the units in them, so maybe not. But we'd be throwing away a lot of our data. So in this case, the better option, what we'd like to do, is to use CryoSPARC to somehow superimpose these 4 copies of the encapsulated ferritin so that they can be combined into one structure for a final refinement. A priori, it's expected that each of these 4 units is identical or indistinguishable. And at least at the scale we're working at now, we don't even have reason to believe that if they weren't, that we had somehow meaningfully separated them yet. So in this case, I felt like this would be a sensible step to do. Now, of course, symmetry averaging that involves a point group symmetry can normally be done via either symmetry expansion or a refinement with symmetry enforced. Now, since the symmetry doesn't follow a point group, particular steps are required to obtain properly symmetry average structures. And the next few slides will show how to do this process, and I'll call this custom symmetry expansion. But before we jump into how you can actually do this in CryoSPARC, I want to make sure that we're all aware, or we're all on the same page as to why we're doing this in the first place. We want to extract as much information as possible from our data, and doing so means that we want CryoSPARC to use all 4 of these units. And we also want it to use the internal 5-fold symmetry in the encapsulated ferritin. So if the 4 encapsulated ferritin units followed a standard point group symmetry, for example, they look like kind of tetrahedral, right? We could simply use a symmetry expansion job, or we could enforce tetrahedral symmetry, and then we could follow that with a local refinement. Now, I want you guys to think again. Why doesn't the arrangement of these 4 encapsulated ferritin molecules follow a tetrahedral group? And what would you think would happen if we did enforce tetrahedral symmetry in a homogeneous refinement of this complex? And so specifically think about the symmetry of a tetrahedron. How many times can you rotate it around this vertex and have it look unchanged? And I probably talked about enough, so I think maybe you guys can just begin to share already. Okay, sorry, I'll do a minute. Maybe that's too much. Yeah, so in a minute I'll call out, and then you'll have time to share with your neighbor. Okay, now you can share with your neighbor any thoughts. Okay, why don't we bring it in? Kind of realizing now it would be nice if we had like 3D tetrahedron foam cubes or something to help reason with or something. Yeah, reasoning in 3D is quite tricky. Okay, so the answer that I came up with was a tetrahedron has around at least the axis shown here in that image, it has threefold symmetry. But each encapsulated ferritin has fivefold symmetry. And so this mismatch is the issue that fundamentally is why we cannot use tetrahedral symmetry. And if you think about it, the number of asymmetric units in this arrangement here is 10 asymmetric units are inside each of the encapsulated ferritin because it's a D5 symmetry object. And then there are 4 of them. So it's 10 times 4, which is 40 asymmetric units in the complex in total. But a tetrahedral symmetry group only has order 12. So there's a mismatch there, which means it's not doing exactly what we think it is. Out of curiosity, I tried enforcing threefold symmetry on the aligned map of the encapsulated ferritin. This was after I got the results, so a bit of a spoiler. But to see what we might get if we accidentally assumed that this configuration had tetrahedral symmetry. And of course, it resulted in a totally uninterpretable map. No clear secondary structure was visible. And then there were also lots of disconnected blobs of density. I also tried running tetrahedral symmetry on the full structure. And that, even refining it, it resulted in the same thing, stuck at low resolution. And totally like all secondary structure was kind of just gone. So at this point, I could reasonably confidently dismiss the idea of it having threefold symmetry. Okay, so now we're going to go into the guts of how we're actually going to do this custom symmetry expansion process. Now, using the Chimera X's Seger tool, I created 4 mask bases. Each of them was surrounding one of the encapsulated ferritin units. And so Seger is actually quite an excellent map segmentation tool. It's actually developed by one of our speakers on Wednesday, Greg Pintley. And it's conveniently built into UCSF Chimera X, called either segment map or Seger. And so using this consensus map, I lowered the threshold value until exactly 4 disconnected density disks were present in the data set. And then one was corresponding, or each of them was corresponding to an encapsulated ferritin. I didn't lower this too low so that any of the encapsulated ferritin like split into multiple disconnected blobs or anything like that, or too high rather. And then I used the segment by connectivity option I used the segment by connectivity option for segmenting. And this worked pretty well for this data set. It pretty much just immediately spotted the 4 disconnected densities and very easy. Then I saved each segment as an MRC file and I imported this into CryoSPARC. And the next step was to convert each of these segment files into the next step was to convert these mask bases, which are now MRC files that I imported it via import 3D volumes, into actual masks. And as Rich said, it's very important that whenever we're using masks for any refinement or classification jobs, we want to make sure they have a soft edge. And so I converted them to a mask via dilation and padding. And I used the volume tools job for this. So in this case study, I dilated the masks only by about 2 voxels, since you can see the 1.0 threshold is shown there. So they're already quite kind of generous. And then I applied a pretty big soft padding of 16 voxels on top of this. Okay, this is what the tree looks like just for the step of mask import and generation. So I had to use 4 volume tools for each of these. And you can see in those kind of slice plots, each of these masks is oriented a little bit differently. They're all covering each of these 4 units. Okay, now just to recap, after this first group realignment step, we now had particles that have been put into register as best as possible, aligned to this tetrahedron arrangement. Now, reconstruction after this point was only reaching about 11 angstroms, or 10 angstroms, so not very high. And this, again, just to also recap, this custom symmetry expansion step was done because it was known a priori that each of these decamers had the same structure. And this step is what is going to allow us to handle the non-point group symmetry that exists in this conformation. Okay, so the 4 masks imported in CryoSPARC correspond to the pink, the purple, blue, and the yellow masks shown in that top panel. The next step we have to do is to apply the mask to each of these decamers and then superimpose them onto each other. This was done via 2 steps. So firstly, I ran 4 volume alignment tools jobs. And they were used to mask each of these decamers and to recenter them at the center of the box. This is illustrated in the second row of images here. So each of these squares corresponds to one volume alignment tools job. And what I did was I applied the mask, I gave it the input mask, I gave it the full tetrahedron decamer, sorry, the full tetrahedron, so all 4 of those decamers, and I also gave it all the particles. Then I turned on recentering. So I said, okay, I want you to apply the mask to this volume and then push it to the center of the mass. So make the center of the center of mass of the volume at the center of the box. And that's what volume alignment tools can do without specifying like a manual center in coordinates. So it's a little bit more convenient to just click, okay, I want to recenter the thing on the center of the mass. And after doing all of this, I kind of got 4 volumes that were centered, but they were not rotationally aligned. And this is shown in that bottom square there. The yellow, pink, purple, and blue kind of all overlaid on each other, but not rotationally aligned. So they're not aligned yet, but they are centered. And the important thing to note as well about this step is that the volume alignment tool's job operated on the particles here. So all the particles were also kept in register at every stage of the process, which was necessary for us to do this. Okay, so after I did this, I used a single align 3D job, and I used that to rotationally align each of the encapsulated ferritin units so that they're coherently superimposed. And this is illustrated in this bottom panel here. So both of these jobs kept all the particles in register with the volumes, and that was actually what allowed us to do this custom symmetry expansion step. And by virtue of the fact that I ran 4 volume alignment tools jobs in parallel with the 4 volume alignment tools job in parallel, that was the actual step that converted a set of 250,000 particles, each image containing 4 encapsulated ferritin, to a set of 1 million particles, each image containing just 1 decamer, or 1 encapsulated ferritin molecule. One kind of detail to note about this step is in the volume alignment tools job, what I needed to do to tell CryoSPARC to consider these images as separate observations is I needed to activate the little parameter called reassign UIDs. And what that does is it tells CryoSPARC, hey, these particle images are actually different from each other. I want to consider them as different particles. If you didn't turn that on, you would find out pretty quickly when you tried to use them all in downstream job, because CryoSPARC would just merge them all into one and be like, what do you mean 1 million particles? You only have 250,000. So it wouldn't work. Yeah. So this step that I did is basically what converted the 250,000 particles to 1 million and did this 4-fold expansion. And at the output of this, we have 4 times the number of particles, all aligned to the center of the box. Okay. So that was kind of a lot of details. And this step was particularly tricky. So I figured right now we can take some questions. And if you guys can understand this, then you have a very good understanding of symmetry expansion, because we basically just did it manually. Okay, so any questions from this slide or the rest of the case study up until this point, I'd be happy to take.
Hi, my question is, it seems you can refine the capsule to a fairly high resolution. Assuming it has some kind of consistency between the capsule and the content inside of it, could you take advantage of the post-prior of that refinement?
Sorry, could you repeat that last part?
Could you take advantage of the post-prior from your capsule refinement? Because it hugely limit the number of poses you want to search.
Yeah, for sure. Yeah, so once we kind of got particles, so actually throughout this whole process, the poses were initially solved with the Encapsulin complex, but they were kind of kept the whole way through. So the particles were always sort of kept in register with the volume. We never started from, once we got the initial structure of the complex, we actually didn't throw away the poses and then start from scratch again. And I guess it would be possible to do that, but you would probably lose. I imagine that ab initio would struggle just a little bit with the fact that you're using subtracted images. Although, I don't know, it would be worth trying, I think. Yeah, I don't know if that answers your question.
I think just to add to that, I think another way to say what Michael just said is that the prior is actually very strong. Like, we are very strongly assuming that each of those 4 pieces of cargo are strongly related in their pose to where they are in the shell, because those poses that initially came from aligning the shell, like the poses from the very first reconstruction on the top right there, those are maintained throughout except for doing small changes to them, like aligning the tetrahedron to itself in different classes and then doing local refinements after that. So we never throw away the information about which of those 4 cargo pieces is where inside the shell. And so that means the prior is actually very strong. And this is an interesting concept because I think Michael also mentioned that the particle subtraction was a key piece here to be able to actually start looking at the internal parts of the shell. But the particle subtraction depends on us having figured out the pose of the shell just perfectly, right? To be able to subtract it. But those images now after subtraction, although we subtracted away the shell, the pose of the shell is now baked into those images because we used the reference of what the shell looks like to subtract it away. If there were in some of the images, if the shell looked a little bit different, that subtraction may not have been perfect. And so in a lot of ways, the poses that we solved at this point are really like baked right into the whole workflow. Yeah. That's a great question.
Good question.
Thanks. Just another thing regarding this long process you resolved the ferritin structure. Because I've heard some concepts like I think it's called local symmetry. So you use a few of operation to move all those subunits together. Is it similar or essentially the same as what you did here?
Yeah. So I am really not a Reliant expert, but I think that they do have a similar, like they have a local symmetry thing where you can specify a set of transformations like rotation matrices and shift vectors to align different subunits. That is pretty much exactly what I'm doing here. In this step here. The reason why I did it this way is just it's a lot of work to figure out, okay, what's the exact shift and rotation like by hand or in Chimera or otherwise that you need to align all 4 of these. And it's kind of nice that in CryoSPARC, because we have Align 3D, we can do this to some extent. So that's why I use Align 3D. But what this step actually does is basically that. It generates a symmetry expanded step where you've applied that local symmetry. I guess I kind of use the term non-point group symmetry more than local symmetry because in this data set, it's kind of there's so much going on that the local symmetry here is what I'm calling the D5 symmetry of each encapsulated ferritin, which we still haven't even applied yet. But yeah, so in that sense, this is kind of CryoSPARC's version of how you might do that in Reliant. Yeah.
So D5 is followed by the tetrahedral is making it more complex, seems to be. D5 and the tetrahedral. For example, D5 in combination with the tetrahedral is making it more complicated.
Yeah, that's
so D5 with any other operations will be easy to process the data. For example, any other point groups C2 or C3.
Yeah, like are you saying like if you had like maybe not this tetrahedron thing, but like you had
instead of tetrahedral, there is some other more symmetric one.
Yeah, some other symmetry mismatch where like
if that is the case, how you are going to?
Well, if you had any other symmetry mismatch, you wouldn't like if you didn't have a non-point group symmetry, you wouldn't have to do this symmetry expansion step. You would still have to deal with the fact that at the beginning of your alignment or at the beginning of your reconstruction, you're probably going to get a map that's aligned to one of like, for example, let's say we go back to.
For example, D5 is a combination with C3.
Oh yeah.
Yeah. So what would be the case?
Do you have an example where like you get a map where it's like the C3 example, the C3 region, for example, is like well resolved, but the D5 is like blurry and incorrectly resolved, or like vice versa? Or
yes, vice versa.
Okay.
Yeah.
Question is about what if you had something other than a tetrahedron with the same D5 symmetry of the actual object?
Right.
But the objects are in some other arrangement besides a tetrahedron, then would there be some case that's easier, or would you have to do this differently if it wasn't a tetrahedron but something else, like just a triangle or something?
Right. Yeah. What you would have to do in pretty much any case is I'll just find it. Actually, I don't have a slide. I don't have a slide showing exactly it. But what you would have to do is you would have to use volume alignment tools to put you would have to realign that portion of your structure that is D5, for example, to the correct symmetry axis. If you have this symmetry mismatch where it's C3 and D5, it's probably unlikely that they will both have the same symmetry axis and be already aligned. So what you would have to do is, like, let's say you got a reconstruction of the C3 part, then you would have to, okay, you can do this particle subtraction thing to subtract away that portion, and then you'll have like particles that have some sort of D5 complex left in those images. Then what you'd have to do is you'd have to take your best reconstruction so far, make a mask around it, and then use the volume alignment tool's job to align that to the D5 symmetry axis. And then from there, I would suggest like doing the rest of what I did in the case study, for example, 3D classification to remove junk, and then try local refinement from there. Yeah. Yeah. You have to get the symmetry axis to be aligned with CryoSPARC's convention, or else it will not work. Yeah.
This reminds me a lot of the symmetry mismatch at the portal complex of a virus, where you have a five-fold vertex and the six-fold or 12-fold or 18-fold of the portal complex.
Right.
In that case, there's a little bit of difference because the five-fold and the six-fold are coincident. Those axes are number one coincident, so it's a little easier to figure this out.
Yeah.
And what usually happens is that you look at the actual touching of the five-fold densities and the six-fold densities, and you see that, yeah, they are slightly different because they're interacting with something a little bit different around each of them.
Right.
And my question here is, it doesn't look like the ferritin is very close to the inside wall of the Encapsulin. So is there really a lot of contact at all between the ferritin and the shell, the Encapsulin? There has to be something, because otherwise it wouldn't be inside at all and wouldn't look like tetrahedron. But it's much different than that kind of symmetry mismatch, which is we've seen a lot of in viruses.
Right, that's a really good point. Yeah. So there's like a big kind of gap between the capsulin and the capsulin region. I pretty much know nothing about the biology of the sample, so I cannot comment on the kind of that. But I did work with I did try at least a virus that had a similar mismatch. It had, I think it was a 5-axis was mismatched with a 3-fold symmetry object on top. And what I tried a lot with that, they just said, was symmetry relaxation, because in that case, you're right, the axes were already coincident, and I was able to, I still have to do this like volume alignment tools to align the symmetry axis with the Z, because it was originally an icosahedral, and so it was in some random orientation. And that did work reasonably well, but I wasn't able, like, that data set was also hard for a lot of other reasons. So, but yeah, I think in that case, symmetry relaxation is definitely useful, because you're specifically looking for, like, okay, if it's like 5 versus 6 or whatever, you'd want to relax with like 6, or maybe even like, yeah, no, I would relax with 6 symmetry, I think, 6-fold symmetry rather. Yeah.
I think in this case, it's interesting that there, like you said, there has to be something that's holding the cargo inside the shell in some like reasonably conserved arrangement for it to even stay in a tetrahedral packing inside. And if it's away from the shell, there must be some linkers or something. But clearly, those parts of the structure have to break both symmetries of the tetrahedron and the D5 and the shell symmetry. So it's a very complicated object, but a super interesting case. Yeah.
Okay, cool. Okay, great. Any other? Yeah, any questions from Zoom as well? Maybe we can.
There are some questions from earlier in the day. One is you may have touched on it earlier about the in the calmodulin case bound to the channel. Is there a way of actually determining what fraction of them have only one, and how could there be two just because of mismatch and misclassification, or could there be, or is there definitely two because sometimes there really are two?
It's a really interesting question, and it's one that I think it's really fun to play with. In this particular case, I just kind of trust the authors to say there's sometimes two, but you can actually do a bit of counting. What you can do is do symmetry expansion, which is that one of the ways of handling symmetry where you actually kind of create n individual copies of these images, right, that are rotated. And then if you do a 3D classification on just one of those calmodulin positions, so you have 4 images that are rotated 0, 90, 180, and 270, and then you classify position A, it's like the same position, yes or no calmodulin, you're really looking at all 4 positions because the particle has been rotated. So each position has been rotated kind of into your mask. So you have a focus mask around a single calmodulin position with a symmetry expanded particle stack. You then have a class that says yes or no calmodulin just inside that one position. But that one position is really all 4. And then you can go through, you have to use CryoSPARC tools for this. And I've written an example online to do this. And you can go through and count the number of times each particle appears in the yes calmodulin or no calmodulin class. And so that will give you kind of I would say it will give you an estimate of how many times there really is a calmodulin bound. Of course, here we're talking about single particle images again. And so you run into this problem. These things are very noisy and very low quality. And so I wouldn't use that to say like, for sure, this particle has exactly 1, exactly 2, exactly 3. But it is a way to kind of approach this from a different angle and try to count kind of how many and classify them out. Yeah. And I think we'll have time for questions on the earlier stuff at the end.
Yeah.
Okay.
Too, so.
Yeah. So I have probably about 5 to 10 more minutes of content just to finish off this case study. And then we can take questions about kind of anything that came out through the day from this case study, anything before. But yeah, let's get to some structures because this has been a lot of data processing. Okay. Okay, so just to recap, from the previous slide, now we have quadrupled the number of particles in our data set. So we have from 250,000 to 1 million particles. And we have superimposed them coherently using a combination of volume alignment tools and the line 3D maps job. Okay, now I wanted to retry local refinement for the second time to see if it works now. And this last step from the custom symmetry expansion was the align 3D job where we superimpose all 4 of them. So I'm showing that up here on the top. Okay, so using local refinement on the full set of 1 million particles, it reached a claim resolution of 5 angstroms, but it's clear this is an overestimate. Unfortunately, we're still seeing some overfitting artifacts, and this is visible in the top-down view at high threshold particularly. So this could be for a variety of reasons. At this stage, though, we haven't actually done any particle curation in 3D. So I picked about 250,000 particles from that initial curation of the 2D classification job, which I didn't show. But those 250,000, I did no curation in 3D. I went straight to ab initio, then refinement, and never did any kind of filtering. And we kept all volumes, of course, from the previous classification job, so they're all still there. And as Rich said, curation is pretty important, and curation in 2D is often not enough to cut it for difficult data sets. So curation in 3D seemed like the best way forward. And so what I did was I launched 3D classification job on the full set of 1 million particles. And taking these manually expanded particles and running a 3D classification now we're finally beginning to get somewhere. And this GIF here is showing all the classes that came out of this 20 class job. Resolutions were improving significantly. And the 5-fold symmetry that you expected to see in the encapsulated ferritin is finally starting to show up. And you can see in some of these classes here, they actually have, it's sort of like, you can see the 5-fold symmetry. And here we actually haven't applied the D5 symmetry yet, just that tetrahedron symmetry expansion stuff. Now, despite superimposing each of those encapsulated ferritin in the previous slide, there's still significant orientation diversity in these classes. And this is shown by this volume series in blue. It's kind of jumping around quite a bit. And the volume that is supposed to be constant in this is actually this big one in the center. So 3D classification found quite a few groups of particles that were still out of alignment with each other. And this gives further support to the idea that this tetrahedral arrangement is just quite approximate and sort of variable in each particle, not constant really. Okay, it is encouraging though that we finally started to see this five-fold symmetry that we expected. And so after one more 3D classification with 20 classes, it was clear that we're beginning to find some conformational heterogeneity as well. So these classes produced by the last 3D classification are shown in this bottom row here in gray. The first 3D classification is in blue, and then the second one was in gray. And you can see from these gray classes that it kind of looked like they were divided into like two main groups. Some of these classes, for example, the top left one, you can't see my, oh, there it is. Okay, these bottom rows right here, they kind of had that five-fold symmetry apparent. And then we saw some classes where it seems like they're broken, or maybe they're misaligned or something, but we probably want to get rid of them if we want to do anything downstream. So to handle this, I chose to use the regroup 3D classes job. And after running regroup 3D, the volumes were divided into two superclasses, and it did a fairly decent job at capturing this qualitative difference that I observed. So superclass 0, which is in purple, had what appeared to be these broken or misaligned encapsulated ferritin classes, where superclass 1 appeared to have the intact five-fold symmetry. And this job was nice because it also put all of the classes back into register as well after they got out of alignment here. Okay, using the two best classes with clear five-fold symmetry from the previous 2D classification, these two right here, I then aligned the structure to the D5 symmetry axis using the volume alignment tools job. This is important because these classes were previously out of alignment. We've never done anything with D5 symmetry yet. So we need to align it to the 3D5 axis before we go on to the next step. And then I finally decided, okay, let's try local refinement a third time. I haven't given up. I really want to see if we can get to high resolution. And now I've removed some junk particles. I've aligned it to the symmetry axes. Maybe it will work now. Okay, so this final refinement was finally able to kick up the resolution all the way from like 7 angstroms where it was stuck before to about 2.8. And so now I want you guys to think, what changed from the initial local refinement before? So why did it work better now? What was different about the inputs that we gave local refinement this time? And which of these differences were important? Yeah, so think for about a minute on this, and then I will tell you to discuss with your neighbor. All right, now discuss with your neighbor. I'll give you guys 4 minutes. Okay, let's bring it in again, and I'll kind of talk about what my thoughts on this were, just based on my experimentation, but it's not, again, probably many reasons, and probably reasons that I didn't even think of are contributing to why this worked better now. So the big difference here that the local refinement was dealing with is that the particles now had more accurate starting orientations. So earlier on in the workflow, I can go back to what the volumes kind of looked like back then, but we were trying local refinement on really this salmon colored volume here. And we were like back at 10 angstroms. Here, it's kind of debatable whether you can even see the five-fold symmetry or not. And certainly the poses are quite coarse, right? Right. So early in the workflow, the orientations were quite poor and likely too far away from their optimal values. But resolving this internal orientation discrepancy using the group realignment was crucial to fix these or improve these orientations. Because here you can see, without even doing anything before, it seems like we've actually started to get the five-fold axis in the right place and the symmetry resolved. Before we weren't seeing that. And so this group realignment step allowed us to put particles back into register before the reference was high resolution to allow doing this on a per particle basis, which is what we hoped the initial local refinement would have done, but didn't. And the other thing that I think was important was that local refinement worked better now because there was minimal heterogeneity. So you can see in this graphic, I'm actually using only 2 of these 20 classes, and that corresponded to about 110,000 particles. So about 10% of our 1 million particle data set. All of these broken and misaligned ferritin classes here were removed. And I also removed a bunch of these particles that were probably fine, but didn't actually result in better refinements than the one that I got with just these 2. So particle stack is much cleaner than before. That's the second reason that I thought is kind of important. These first 2 points are probably the most important, so better orientations and cleaner stack. As we talked about, local refinement is pretty sensitive to junk particles. The algorithm will really kind of look for any opportunity to give it to overfit in the case where your masks are small. And so it's pretty crucial to do as much particle curation as possible before jumping into local refinement. And that's typically our suggestion when people see artifacts in the map or they're not getting to high resolution. Yeah, and so if particle alignments are too far off, you can somewhat attempt to work around this by increasing the search extents in local refinement. But as we talked about before, that increases the odds of you overfitting. Okay, and then the other reason why we got better resolution now is because we finally enforced D5 symmetry. So of course, it's going to give us high resolutions. But in this case, it didn't seem to be artifactual. And here's just a quick comparison of the density map that we recovered. And so this density map in gray, this one was deposited, but it was recovered without applying D5 symmetry. So this is not a fair comparison at all. But it is what this kind of shows to me is that doing all of this intricate geometry work and using 3D classification to remove as much junk as possible can actually have pretty big impacts on getting a better structure. Because we removed the junk, we're giving local refinement better orientations to start out with, and we're finally getting the D5 symmetry aligned with the correct axis and then imposing it. Okay. And that is pretty much all I wanted to talk about today. Yeah, thank you very much for your attention. I also want to give a big thank you to the CryoSPARC team and Rich and Ali as well. Yeah, now we have about 10 more minutes left for any questions from the rest of today. Sorry, from the rest of this case study or from earlier in today. Yes.
One thing about this encapsulated ferritin is that in almost every image of the 4 objects that are inside the encapsulated icosahedron,
yeah,
almost always several of those are overlapping.
Right, like the encapsulated ferritin.
Yeah, this ferritin and the next ferritin are overlapping.
Right.
Is it possible that the ones you picked, yeah, almost all the time, the ferritins are going to be on top of each other.
Yeah.
And it's very, very seldom that you'll have one sticking off on the side it's all by itself. Is it possible that if you looked at the orientations of those 110K things that finally gave you the right answer, is that they were all kind of non-superimposed on each other?
Right. Yeah. It's a really good question.
That's just something that everyone worries about. Oh, are our particles overlapping? Well, almost all of them are.
Yeah.
Maybe not. I don't know.
No, that's a good point. My guess is, and okay, I can actually take a look. One thing that I want to look before I try to answer that question is, oops, cursor's here. So this was a job that I ran, and the orientation distribution is pretty uniform. For like, I don't know, it's not, if it was what you were saying, I think what you would see is a lot of like side views, for example, like views where you're looking along the side of the ferritin, but if it was a top view, it would be like occluded on top of some other ferritin, right? But in this case, like if that was the case, we'd see a bunch of particles like along this band, along the zero elevation band, but like not that many along the top and the bottom. So my guess is no, but yeah, my guess is no. Yeah. Yeah, but it is interesting that like the overlap didn't pose that many problems. I think before we went to the single ferritin, it was fine because we were still modeling the whole tetrahedron. But you're probably right that we could do even better results if we like, now that we have this aligned structure, we went backwards and we subtracted it from all of the other particles. But my brain like hurts when I even think about that. It could improve things, but I was like, okay, we got 2.7, it's fine. But yeah, good question.
Yeah, I have a question here. Right here.
Yeah.
Yeah. It's a very interesting assembly questions of these particles. And on the icosahedral particle, I expect that will be correct because you get 2.8 angstrom, they can build model. Do they know what kinds of molecules that make up the tetrahedral particle inside? Is that a protein?
So I admittedly know very little about the actual biology.
So based on your slide, it stated about 5.5 angstrom at the end. Is that correct?
Oh, yes.
Okay, at 5.5 angstrom,
the cargo goes to 2.8 angstroms at the end, and it's ferritin of some sort.
No, the one that you showed, the 2.8.
Yeah.
Is a icosahedral.
No, that's the cargo that was
the cargo inside.
Yeah.
Okay. So what is the 5.5 angstrom thing?
That's just a published map from this data previously.
Yeah.
So the same data, the people who originally published it, they only got to 5.5 processing the data with a different workflow.
I see.
Whereas this workflow gets to 2.8 with the steps.
2.8. Okay. So you can probably build the model from that.
Actually, Michael, do you want to show the model building?
Yeah. So there's actually one kind of thing that I skipped over for time, but the initial, so this structure had a I should say I should not get this wrong. There was a similar crystal structure on the PDB that the authors originally kind of used to compare their structure to. What we did at the end after we got this atomic map was we took the PDB entry that this atomic model is from, and we re-refined it into our density map that we attained from CryoSPARC. And then this atomic model was from E et al. From 2019, and this was from X-ray diffraction. And this was like 2.1 angstrom map, so it's much higher resolution, or significantly higher resolution. Yeah, so what we were able to do was we kind of re-refined the sequence into our density map, and then we got the atomic models shown in panels C and D. And we were able to find like these extra residues, but we weren't really sure about whether it was biologically relevant or anything like that. But yeah. So originally, this gray map here was from this was from the cryo-EM density, the cryo-EM, the same data. But they didn't do like that
So is that
non-group
right? The one
stuff.
on the gray color one was the published one?
Yeah, it was published, but it wasn't like referenced in the paper. It was published, and then they didn't like claim it was. They didn't make any claims about the structure, I believe. Yeah.
So it's possible the original authors may have been able to do better if they had also enforced D5 symmetry and etc. But this was just a comparison to show from the same data what we could get from inside.
In other words, if you do it right,
Yeah.
you'll see more.
Well,
That's
as
what-
best as we could figure out.
Maybe
yeah.
that's much easier to digest.
Yeah, for sure.
Okay. All right. Thank you.
Hi.
Hi.
I'm curious on because this is more of a biological question. How to interpret this data? Assuming you can find some tetramers that has all the good particles, is there exists a set of full particles and then their respective poses that can reconstruct you the full map and to see what's going on there? If not, because I do wonder this 2.8 angstrom map, other than protein identification, whether it can provide us more information.
Yeah, that's a good question. Like, I guess one thing that you're kind of asking is like, do we expect this tetrahedron arrangement to exist in all of the like particles, for example, or like how can we interpret? Because we've gotten a good structure of just one ferritin molecule, right? But like that's not in the context of the broader tetrahedron arrangement. We don't know what that looks like. And it looks like there's a lot of heterogeneity.
Let's say, for example, I want to see the contact point between the ferritin and the capsule.
Yeah. So I mean, to my knowledge, that wouldn't be like a question you could answer with this map. Yeah, or at least with this, with what I've done in this case study. Yeah.
A question from the Zoom people. How can you resolve small differences in amino acids? I have a heteromeric assembly of proteins, which is almost similar in sequence, but only differs in 1 or 2 amino acids in one helix. The data was processed in C1 so as not to mask these small differences. However, these differences are not quite distinguished, even if the local resolution is 2.5 angstroms or better. What can you do?
That's a very good question.
That's a hard one.
I can start with this one. That's a very challenging problem. At the end of the day, I think one of the themes that you can take away from learning all of this stuff is that, and Rich kind of said this at the beginning, cryo-EM is all about the images. Every single particle image only has so much signal in it. And if you want to be able to make distinctions like, is this subunit type A or type B, and the difference between those is only a few amino acids, you're asking the question, does a single individual particle image have enough signal to be able to distinguish these two objects? And if those two objects are so similar that in the image with all the noise present, there's absolutely no way to tell them apart, that's your answer right there. You cannot tell the difference between version A and version B if there's such a tiny difference. The only hope you have is if that tiny difference also somehow, because of your chemistry or protein structure, whatever, is magnified into a larger conformational change across the entire structure. Even if that's just half an angstrom displacement of a whole bunch of atoms, if there's a ton of atoms that are in a slightly different place because of one amino acid difference, that becomes detectable because now that the difference in the image between type A and type B suddenly becomes much larger. Even though it's a very minute like atomic displacement, it becomes detectable. So that's really what it comes down to. It's a similar question people ask, like, oh, can I detect the presence of a ligand or not? If the presence of a tiny ligand, just a few atoms, is the only difference between two versions of an image, you can see for yourself that the cryo-EM images are not going to support that kind of fine ability to distinguish two states. But if the presence of the ligand also induces a tiny conformational change across the protein that moves a bunch of atoms just a little bit, that becomes now more detectable. So that's like
that's great. And I just wanted to point out also that that's a big difference between resolution in something like X-ray crystallography and resolution in cryo-EM, where in crystallography, I think it is much more true that if you have a 2 angstrom map, you're getting this like really sharp contour, and you might see a blur where this residue has this occupancy, or there's these different conformations, right? In cryo-EM, you can have, our resolution is a measurement. It's when we say resolution, we're talking about reliability, right? Up to this frequency, the half sets are the same. And so you can say these half sets very reliably have either an alanine or a threonine in this position. And, you won't be able to resolve that using that map, like Ali is saying, because there's just not, these images are just too bad for that to happen. And in something like X-ray, your data, your raw data, those reflections are already averaged across the millions and millions, more than the milligrams of protein, right? So I think that's the big challenge with something like that. Yeah.
I'm just curious, if you take on a project like this a priori, and all you see is these little peace signs on your grids, how do you know what kind of symmetry to impose? In crystallography, you do a self-rotation function and figure out where all the symmetry centers are.
Right.
But how do you know to impose the symmetry that you need to impose to get, the 2.8 angstrom or 2.5 that you showed later?
Yeah.
From the start.
Yeah, that's a good question. I think the most the hardest one to find out was the D5 symmetry, at least in this case. The answer for me was that it was known, or it was described in the paper. But if I was processing this from scratch, the tetrahedral arrangement thing, that would have to be fixed somehow, or at least modeled somehow. So that was what kind of led to the impetus of doing that group custom symmetry expansion step. And then actually going from that to realizing that there's 5-fold symmetry on these encapsulated ferritin. In general, it's hard to do. I think where it would have been most clear is when you get to this point where you can kind of see it in your structure here. At this point, I was like, okay, there's probably a subset of particles where it is an intact 5-fold symmetric object. Then the question is it C5 or D5? So again, it's kind of hard to answer in general.
Actually, a follow-up there, Michael. I'm not sure if you tried this, but did you try a C1 refinement of the same particles that went into your final structure? Like on the next slide, you have those 2 best subclasses. Assuming you were doing this with no prior knowledge, you might get to this point where you're like, okay, I just want to see what is all the heterogeneity in these things, and then you start to see these 5-fold symmetry versions show up. Then your first next step might be to just try and refine those particles in C1, not assuming anything.
Yeah,
I'm not sure if you tried that.
Yeah, that's a really good point. Yeah, so I didn't try that because I think I was convinced that there was D5 symmetry from the paper. But actually, that would be a very good point, is to just don't impose symmetry, don't kind of bake in anything at this point, because we've gotten so far without doing anything symmetric yet. And then let's say without D5 symmetry, okay, you're moving 10, but you're moving 10, you're decimating the number of particles you have. But presumably you could have gotten to decent resolution without that if you gave them the same particles, just not enforcing symmetry. So that would be a good step to do, for sure.
Just as a guess, the same particles in C1 might have got to 3.8 or 4 angstroms or something like that, enough to just see that, okay, there's definitely a clear repetition of subunits here. And then your other biochemical knowledge about what this protein probably is might tell you something, or I don't know, mass spec and other things, or whatever helps you count the number, like the stoichiometric ratios might help at that point. But it is fair to say that a workflow like this in real life is going to be very exploratory, and it's always good to just try more stuff, because you might find that your data is actually good enough to get this, even though you may not suspect that at the beginning. I think another point that Michael raised early on that's worth reiterating is that a great thing about a data set like this is that right from the beginning, you know that the quality of the images is actually really good, because you're able to get the shell to a very high resolution without doing much. So you know that the actual exposures, motion correction, CTF estimation, all the other stuff that could have gone wrong is actually good. So now it's worth spending the time to figure out all the details of particle alignment, because if you know that if you were to do that well enough, there's a good chance you'll get a good structure, since the signal quality is actually really good in the images.
Yeah, it's a great point.
One last question, please.
Yeah, I think we're about at time.
So my question kind of follows up with the question beforehand about amino acids and resolvability. And and whether or not, so if you have a knock-on effect that changes a whole bunch of atoms, you can distinguish that real easy. What do you think about the development of laser phase plates? Because really we're talking about a signal to noise ratio problem, right? So you add a laser phase plate, how does that change the equation?
I am probably not the most familiar with phase plates.
I can start
Ellie can
on
take
that one. So phase plates in general are definitely something that, from an image processing perspective, theoretically would be amazing and awesome, especially for tomography, but we can leave that as a separate topic. I'm sure with the laser phase plate, there are issues about actually building one and having one in your microscope that are a whole different can of worms. But in principle, if such a thing were to be able to be created and robustly used in image acquisition, it would make a big difference in a few ways. It's unclear how much difference it would make on the high resolution end. Like if you're already able to prepare a sample of a target, collect data the standard way and get a high resolution structure, that whole story may not change dramatically with the faceplate. The biggest differences will be in alignability of smaller things. So going down to smaller size scales with Cryo-EM, because the faceplate fundamentally changes the CTF and allows us to get low resolution contrast much better than defocus can. But in practice, you have to combine defocus with your faceplate, because at the end of the day, you still need a really good CTF estimate to be able to correct the high resolution CTF. And you can't detect the CTF unless there are tone rings. So you have to be out of focus a little bit. Anyway, that's a whole separate story. But the big improvements would be in aligning small things. The second one would be in being able to sort out heterogeneity at a smaller and smaller scale, like smaller pieces of a larger thing moving or changing, appearing, disappearing, which now become more apparent with better low and medium resolution contrast. That's really the hope. Like that would be the dream, I think, at the current moment for single particle for faceplates.